Welcome back everyone to this biochemistry lecture. Today we are going to start this discussion on a new topic in biochemistry, that is our amino acid metabolism. Okay. Previously we have discussed carbohydrate metabolism, glucose, glycogen along with pyruvate dehydrogenase. So let's start this amino acid metabolism. First of all, we will start with general amino acid metabolism. So this is a general structure of amino acid. It's in double A is for amino acid. Okay, so we'll draw a general structure for amino acid. This is the center of carbon, this is the hydrogen group, this is the COH group, this is the NH2 group, and here is the R group. R group could be whatever different. This carbon is our alpha carbon. Okay, this is the first reaction we are discussing. Okay, now this is decarboxylated. Okay, this amino acid is decarboxylated, so will form this compound, so alpha carbon. Okay, this compound will be formed. This is biologically important amine. Biologically important amine. Okay, now for this decarboxylation, enzyme for amino acid decarboxylation is PLP, pyridoxal pyrophosphate. Pyrophosphate pyrophosphate or phosphate, pyridoxal phosphate, PLP. These are coenzyme. The decarboxylase enzyme and the coenzyme is our PLP. Okay, the first reaction. Another reaction, first here we have removed our carbon dioxide. In the second reaction we are going to remove the amino group. Okay, this is our NH2, so our R group. Okay, NH3 is removed, so we are left with COOH, H, and here an oxide bond is formed. Okay, this is our alpha keto acid form. On deamination, alpha keto acid group is formed. This is our deamination reaction. Deamination reaction. So we have seen two basic reactions in which the two most important functional groups of an amino acid removed. First COH, CO2 is removed and amino group is removed. Biologically important amines and alpha keto acids are found. So let's start our proper discussion with transamination. Transamination. Okay, we'll start with our discussion. Definition. The definition is transfer of amino group from one alpha amino acid to a alpha keto acid. Amino group transfer from one alpha amino acid to alpha keto acid to form a new pair of alpha amino acid and alpha keto acid. Ka for keto acid, A for is for amino acid. So from one alpha amino acid, an amino group is transferred to an alpha keto acid. In result, a new set of alpha amino acid and alpha keto acid are formed. New pair of alpha amino acid and alpha keto acid are formed. So alpha amino acid, alpha keto acid. Okay, this is the old form, and in the new form here Upon transfer, alpha keto acid and alpha amino acid are formed. Basic transamination formed reaction. Okay, this transamination takes place in all organs. Okay, in cytoplasm of all organs transamination takes place. Ammonia is not released freely. Ammonia is not released freely. It's a reversible reaction. Coenzyme here is PLP, pyridoxal phosphate. What is this? B6 active form, butane B6 active form. Okay, throughout this discussion of our amino acid metabolism, different amino acid metabolism, that is branch chain or anything amino acid metabolism, PLP will be the major coenzyme. Coenzyme is our PLP, pyridoxal phosphate B6 active form. Okay, remember this. So we will see some examples of transamination. Two important examples. First is our involvement of alanine aminotransferase. Alanine aminotransferase, ALT. Okay, it's also called SGPT. Okay, here first set we have alanine getting converted to pyruvate. Also seen this reaction in pyruvate dehydrogenase discussion, go check that one out. And in correspondence, here this is one alpha amino acid, this is one alpha keto acid, here what will be formed? Alpha ketoglutarate. These are alpha keto acid, along with glutamate will be formed. Glutamate, a new pair of alpha amino acid. So alpha amino acid dash or prime. Okay, transfer of amino group from one alpha amino acid to one alpha keto acid to form a new pair of alpha amino acid and alpha keto acid, alpha ketoglutarate and glutamate, and alanine will get converted to all important pyruvate. Okay, another example we will see here, solidifier foundation. Involvement of aspartate aminotransferase. Aspartate aminotransferase. These are AST. So called SGOT. Okay, aspartate aminotransferase. That was alanine aminotransferase. ALT or SGPT, AST or SGOT. Okay, here we have involvement of alanine. Sorry, aspartate is involved here. Aspartate is going to be converted to oxaloacetate. Here is the enzyme is ALT. Okay, here the enzyme is AST. Correspondingly, the opposite side, a new pair of alpha amino acid and keto acid will be formed. This will be same as before, alpha ketoglutarate and glutamate. Okay, alpha keto acid new, alpha amino acid new. So alpha amino acid from which amino group is transferred to alpha keto acid. So we've seen that this part of the reaction, the left side of this reaction is different. This part of reaction is different. This side of reaction is the same. Okay, so what does this explain or mean? Let's find out. So we've seen that this part here is specific, this part here is non-specific. Okay, so we can also say that transamination is specific for one pair of substrate and non-specific for the other. Non-specific for the other. Okay, so we can say the general reaction, general transamination reaction. What can we say finally? That any alpha amino acid, any alpha keto acid transfer amino group will always lead to formation of alpha ketoglutarate and glutamate. Okay, this is the gist of our transamination reaction. Again we will see the reactions. Involvement of ALT, alanine amino transferase, alanine converted to pyruvate. ALT is also known as SGPT. AST, aspartate amino transferase, aspartate to oxaloacetate. AST is also known as SGOT. And this alpha ketoglutarate and glutamate are remaining same. Okay, yes. So moving on. There's a concept that transamination as a funnel. Transamination as a funnel. Okay, what do we mean by this? Okay, so as we said, a funnel, so we'll draw a funnel first. This is the opening of the funnel, this is the discharge route. So this is a funnel. Okay, in this funnel alpha amino acid is getting introduced and coming out as alpha keto acid. Alpha amino acid could be alanine, alpha keto acid could be pyruvate, or it could be aspartate and oxaloacetate. Okay, now here alpha ketoglutarate is getting converted to glutamate. Okay, glutamate is coming out of this funnel. This funnel is our transaminase funnel. Transaminase funnel. So we can say that alpha amino group of any alpha amino acid is getting concentrated as glutamate. Okay, so we can conclude that glutamate is the only amino acid, okay, that can undergo oxidative deamination. Oxidative deamination. Okay, glutamate is the only amino acid that can undergo oxidative deamination. Very important point, glutamate. Okay, now moving on, we have our this transamination is required for biosynthesis of non-essential amino acids. Non-essential amino acids. Okay, biosynthesis of non-essential amino acid. For example, alpha ketoglutarate may be converted to glutamate. We have seen this. Pyruvate can be converted to alanine. Okay, oxaloacetate can be converted back to aspartate. These are our non-essential amino acids. non-essential amino acids. Okay, so you've seen all this here, that alpha amino group of the alpha amino acid is getting transferred. Okay, now we'll see an example where the non-alpha amino group undergoing transamination. Okay, for example, here now the non-alpha group of delta amino group of ornithine. Delta amino group of ornithine. Delta amino group of ornithine. The enzyme is delta ornithine aminotransferase. Delta ornithine aminotransferase. Delta group of ornithine, delta ornithine transamination aminotransferase. Delta ornithine aminotransferase is the enzyme here. Clinical importance, we have something here. Clinically, deficiency of the above enzyme. Deficiency of the delta ornithine aminotransferase. Delta ornithine aminotransferase deficiency will lead to gyrate atrophy of retina and choroid. Gyrate atrophy of retina and choroid. Okay, retina and choroids with gyrate atrophy due to deficiency of delta ornithine aminotransferase. And delta amino group of ornithine undergoes is the non-alpha amino group undergoing transformation. Okay, so this was the gyrate atrophy of retina and choroid. Now we'll see some sources of ammonia, where the ammonia is coming from. Some sources of ammonia. Sources of ammonia. Okay, sources of ammonia. First is our alpha amino group amino acid. Alpha amino group amino acid such as glutamate. Glutamate is the only amino acid that undergo oxidative deamination. The next is our non-protein nitrogenous substances, NPN. Non-protein nitrogenous substances, NPNs. Okay, then your amino sugars. From amino sugars you can get ammonia, one of the sources of ammonia. And the last and final is our all important gut bacteria. So glutamate and other alpha amino group amino acids, NPNs, non-protein nitrogen substances, amino sugars, and gut bacteria. Okay, these are the sources of ammonia. Then we'll see how ammonia is getting transported. Transport of ammonia. Okay, transport of ammonia. First we'll go one by one route. First case we're going to see most organs. Okay, including brain. Okay, most organs including brain. How ammonia is getting transported? So we have our alpha amino group, glutamate. Okay, and also other sources, ammonia. Okay, is getting added into here from glutamate along with the source of ammonia getting added here. The enzyme here is our glutamine synthase. glutamine synthase enzyme, and here we have our glutamate, and ammonia from other sources in the presence of glutamine synthase get converted to glutamine. Here one single ATP is used. Okay, how is getting formed? We'll see the structural conversions. This glutamate we have here is simple structure here. This is our glutamate. Okay, this part is getting removed. Okay, this is getting removed to form this. Okay, this part of glutamine. Okay, COOH2 is getting formed. This is our glutamine. Okay, so glutamine is the transport form of ammonia from most organs including brain. Glutamine is the transport form of ammonia in most organs including brain. Enzyme glutamine synthase. Synthesis requires ATP. ATP is getting converted to ADP. Okay, so obviously it's an energy requiring process. It is an energy requiring process. It takes place in mitochondria. It takes place in mitochondria. Okay, it's also called the first line trapping of ammonia. First line trapping of ammonia. Okay, now here the question arises that why ammonia is toxic to the brain. Ammonia is toxic to the brain. Why ammonia must be handled with care. Okay, so we'll see the reason here. Suppose this is a from the brain here, part of the brain here. Okay, this is our blood brain barrier. Blood brain barrier. When ammonia outside is in excess, ammonia outside the brain becomes excess, gets converted to ammonium ions. Ammonium ions. Okay, now this ammonium ions is impermeable to blood brain barrier. This ammonia is in excess is permeable. Ammonia in excess is permeable. Is permeable to blood brain barrier in excess, not in general. In general it is going to be converted to ammonium ions, but in excess it is permeable to blood brain barrier. And here along with glutamate converted to glutamine, presence of glutamine synthase. You know this process before, glutamine synthase. Okay, so in excess of ammonia there is increased glutamine in brain. Okay, increased glutamine in brain. Okay, this glutamine attracts water into brain and cause to brain and cause cerebral edema. Cerebral edema. Glutamine attracts water and causes cerebral edema. Glutamine in brain attracts water, causes cerebral edema. This is the reason why ammonia is very toxic to the brain. Also, this is the first reason. Okay. Second reason is glutamine in the brain undergoes decarboxylation. Glutamate in brain undergoes decarboxylation, and we know glutamate on decarboxylation forms GABA, gamma aminobutyric acid, a depressing neurotransmitter. We all know this GABA is a depressing neurotransmitter. It is formed by decarboxylation of glutamate. Okay, causes CNS depletion, causes CNS depression. Okay, as a result of this formation of glutamate and further conversion to glutamine, alpha ketoglutarate is depleted. Alpha ketoglutarate is depleted and decreased tricarboxylic acid cycle, TCA cycle. So there is decreased synthesis of ATP. Decreased synthesis of ATP. Okay, so these are the three reasons why ammonia is very toxic to the brain. Ammonia in excess gets into the brain, and from glutamine is formed through glutamine synthase. Now this glutamine attracts water and causes cerebral edema, and this glutamate also undergo decarboxylation to produce GABA, causing CNS depression. And alpha ketoglutarate getting depleted, decreased TCA cycle leading to decreased ATP synthesis. So for all these reasons, ammonia should always be handled with care. We have seen how ammonia is getting transported in most organs including brain. So this was our glutamine enzyme, glutamine synthase from glutamate. Now we'll see in muscles, transport ammonia from muscles. From muscles. Okay, from muscles. From muscles, in muscles we have this alpha amino acid getting converted to alpha keto acid, and along with this alpha ketoglutarate and glutamate is formed. Okay, now we all know this is a reversible reaction. Okay, now this glutamate is getting converted to alpha ketoglutarate, along with we know pyruvate. We have pyruvate in muscles, is getting converted to alanine. Okay, this glutamate is getting converted back to alpha ketoglutarate, as this transamination process is a reversible reaction. Along with this pyruvate is converted to alanine. Okay, being converted to alanine. This alanine is the transport form. Transport form of ammonia in muscles. Transport form of ammonia in muscles. What is this? Alanine. Okay, very important discussion. We've seen transporting of ammonia most organs including brain, that is glutamine, and also alanine in muscles from pyruvate. Okay, now we'll see the final disposal of ammonia. Final disposal of ammonia. Final disposal of ammonia. Okay, where this final disposal of ammonia is taking place in liver. First happens it's in liver. Okay, I will see step by step what is happening. This is our liver. Okay, from other organs, what is coming? Glutamine from glutamate. Okay, now this glutamine in the presence of this enzyme glutaminase, okay, is getting converted further back to glutamate, and it enters. Glutaminase enzyme action forms glutamate from glutamine, okay, which enters the liver. Here one water molecule is consumed to ammonia. Okay, now from muscle we have alanine. We have alanine in transport form of ammonia in the muscle. This enters the cycle. Alanine. Okay, and we know glutamate getting converted to alpha ketoglutarate, and alanine is getting converted to pyruvate. Okay, this alpha ketoglutarate is formed from glutamate. This is the most important compound here, glutamate. So finally we can write that everything gets unpacked as glutamate. Why? We have already said before, glutamate is the only amino acid that can undergo oxidative deamination. Only amino acid to undergo oxidative deamination, glutamate. Very important discussion. Everything gets unpacked. Everything gets unpacked as glutamate. This is the only amino acid to undergo oxidative deamination. This is the reason here. These are the steps here. Glutamine and alanine comes from different sides. Glutamine from other organs in brain, alanine from muscle. Action of glutamine is converts to glutamate. Glutamine enters the liver and gets unpacked. Along with this, it gets converted to alpha ketoglutarate. The alanine also forms the pyruvate. This is the gist. What happens at the end to the transport of ammonia? Final disposal is in liver. So we have seen two enzymes from glutamate to glutamine synthase, from glutamine to glutamate, glutaminase. Okay. Now further question remains, what happens to the glutamate? What happens to glutamate? What happens to glutamate? Okay, now this glutamate here, everything gets unpacked as glutamate. Okay, glutamate here is converted to alpha ketoglutarate. We have seen that before. And one ammonia is released. Now this ammonia is released freely. We have seen in transamination that ammonia is not released freely, but in this final disposal of all the transporters of ammonia, everything gets unpacked as glutamate and ammonia is released here freely. Okay, and also NAD plus with phosphate converted to NAD plus H. Okay, this ammonia released enters the urea cycle, which we will discuss in further lectures. The urea cycle and their importance and their disease. This ammonia released freely here, and conversion of glutamate to alpha ketoglutarate enters the urea cycle. Okay. first the enzyme here, glutamate dehydrogenase. Glutamate dehydrogenase. Glutamate dehydrogenase. Okay, this is the enzyme GDH. So above it's a deamination reaction and also oxidation reduction takes place. So deamination along with oxidation reduction. So overall this is our oxidative deamination. Oxidative deamination. Here one hydrogen is getting added, so it's an oxidation reduction process. This is getting reduced, this is getting oxidized, okay, and ammonia is getting released from glutamate, so deamination. So this here is oxidative deamination, ladies and gentlemen. Okay, enzyme is glutamate dehydrogenase. So the enzyme taking part in oxidative deamination is our glutamate dehydrogenase. Finally, alpha ketoglutarate is converted, getting formed from glutamate. Ammonia is released freely here, this step, entering the urea cycle. Okay. Now the features of this oxidative deamination. We'll see some of the features. Features of oxidative deamination. Takes place in liver and kidney. Takes place in liver and kidneys. Okay. Second feature is a reversible reaction. This reaction can be reversed. Very important point. It's a reversible reaction. And the glutamate dehydrogenase is the only enzyme which can use either NAD plus or NADP plus. Only enzyme that can use either NAD plus or NADP plus. Okay, the organelle here is mitochondria. Organelle here is mitochondria. Okay. Lastly, we want to see some of the glutamate dehydrogenase is the enzyme. It's allosteric activator. That is ADP. Okay. Its allosteric inhibitor is ATP along with GTP and NADPH, NADH, NADH. Allosteric inhibitors are ATP, GTP and NADH. Allosteric activator is ADP. So this was all about our discussion on general amino acid metabolism, sources of ammonia, how ammonia is transported in different forms, what happens in liver, the transamination reaction, the ALTs, the ASTs, how transamination acts as a funnel here. Everything is discussed. It's in the general amino acid metabolism. Next we'll be going to our all important urea cycle. So revise this topic, guys. Thanks for watching. I will catch you in the next one. Good luck.